So we have talked about the theory of evolution, and we've backed it up with examples, and we've shown how it's possible for new mutations to arise within an individual. But what we want to talk about now is how does that mutation go from a single individual to the recognition of a new species, right? The accumulation of so many different adaptations that we recognize it as a new species. So today we're going to talk about the biological species concept, a way to define a species, and we'll look at how genetic divergence contributes to this, as well as different modes of speciation of allopatric and sympatric speciation, and different reproductive barriers that help to maintain the species. So we're going to start off by giving a definition of what actually is a species, kind of what constitutes a species. And the biological species concept is one of the most widely accepted definitions of a species. And so it defines a species as individuals that are able to mate together or reproduce together and produce viable and fertile offspring. So members that are of the same species are able to reproduce, and they are producing viable, meaning the offspring are born alive, and the offspring are also able to reproduce themselves. All right. So if we think about this, this is the main way in which we interpret individuals as belonging to the same species. So what we need to think about, though, is does this apply to all species or all organisms, we should say. So there are instances where some organisms don't really sexually reproduce. It's not their main method of reproducing. So that can be very hard to kind of define them using this biological species concept because it depends heavily on who they are mating with. So we see that here it says the biological species concept does not always apply, especially to asexually reproducing organisms. So these are organisms that basically are cloning themselves. They are a single individual that is passing on their genetic information. And this can occur with plants, right? You might have a house plant where somebody kind of broke off a piece of that house plant and gave it to you, and you've rooted it and stuck it in the ground, and now you have a new plant. The same thing can occur with fungi. They might fragment part of their bodies, and now instead of one fungus, there are two of them. So that's asexual reproduction, where you only have one parent that's involved. And so therefore the individuals are not mating, they're not finding a partner and swapping gametes between them. So that makes it very hard to define them using the biological species concept. There are even some animals, such as fish and lizards, that also reproduce asexually sometimes. Now, what about if we're trying to identify organisms just from the fossil record? So all you have is a fossil of them. You don't know what they did during their lifetime. You don't know who they mated with during their lifetime. So obviously, this definition is not going to work for them either. It's going to be very hard to define them using this method. So even though it's kind of the foundation of how we look at things, it doesn't always apply to all organisms or in all cases. So sometimes we have to rely on other ways to identify a species. So for the fossils, we might look at biogeographic information. We might look at where a fossil is found in relation to other known fossils and see, were they in the same region at the same time? And therefore, it could potentially be of the same species. So comparison of where we find them. Now, we can also look at the DNA sequence, right? That's kind of your ultimate definition right there is, do they have the exact same DNA sequence, right? So that would definitely tell you if they are of the same species, or if there's enough differences between them that they should be of a different species. We can also look at morphology. So morphology refers to the physical appearance of the organism. So what do they look like? Do they look very similar to each other? Now, this is what was traditionally used to define species, but it's not always the most reliable. So we can look at, let's say, tree frogs. And so tree frogs belong to the genus Hyla. And there's Hyla crucifer, which is the spring peeper, and Hyla andersoni, which is the pine barrens tree frog. So Hyla crucifer has a little cross shape on its back, and Hyla andersoni has a little lavender stripe that runs down its side. So because they look different, we can tell the different species of the tree frog. So they both belong to the same genus, but they're different species, and they look different from each other. So sometimes that works, sometimes that doesn't work. Sometimes with birds, it's very hard to tell different species apart. And of course, the ability to interbreed, as we already talked about with the biological species concept. All right, so hopefully between some of these methods, we're able to differentiate between different species. So how do we bring about new species? Right? How does that come about? So a lot of times we see kind of two different lines of evolution that have given rise to what we call speciation, which is that process by which a new species is produced. So let's say, for instance, that species A transforms gradually over time to species B, and then usually species A becomes extinct. So that's one way it can go about. And a second method, or second pathway, the parent population, let's say, doesn't go extinct. So a new species evolves, let's say, from species A, we get species B, but species A does not go extinct. So the population kind of buds off of it. And now you have two species instead of one. This is much better for the environment because this creates greater diversity of species, and it's believed that most species have kind of evolved this way. Now, when species are evolving, the crucial part in the kind of birth of a species or the origin of a species occurs when the gene pool, in other words, referring to all the genes that are present in a population, when that gene pool is cut off, or whether it's severed from the population of the parent species. And therefore, we no longer have the exchange of genes, or really the exchange of alleles through gene flow between these two populations. So this can be due to geographic barriers, ecological specialization, natural selection, or behavioral differences. Either way, this isolates the gene pool, and we call it genetic divergence. All right, so evolution can lead to new species when your gene pool becomes severed, right? It becomes cut apart or cut off from each other. And there's no gene flow. Remember, there's no immigration or emigration between the populations. The populations become isolated. And once they're isolated, we can see new mutations starting to pop up in one population and maybe not the other. The other population might be subjected to genetic drift. Remember, genetic drift is just chance events that happen. And so over time, we'll see what's called genetic divergence between these two populations. Right? So there's that accumulation of differences in the DNA sequences. And that could be from chance events, that could be from new mutations that arise and natural selection working on those mutations, so that these two populations now become are very different from each other over time. All right, so these factors can cause a new population to evolve and no longer resemble the population from where it came from. So this isolation from the parent population to bring about speciation can occur because of geographic barriers, reproductive barriers, or ecological isolation, as we'll talk about next. Okay, so one of the most common ways that new species is brought about is from geographic isolation. So geographic barriers such as mountains, rivers, oceans, or even expanding deserts. I think there's actually deserts in China that are expanding, and they're trying to plant a lot of trees to keep the deserts from expanding. They can physically separate a once connected population into two or more isolated groups. Now, once separated, these groups no longer exchange genes because the individuals cannot cross the barrier to mate. So with gene flow cut off, each isolated population experiences its own evolutionary pressures. Once again, different climates they could be experiencing, different predators, food sources, and once again, what we're talking about with genetic drift, all of causing their gene pools to change independently. So over many generations, these genetic differences accumulate, leading to traits or behaviors that prevent successful interbreeding. All right, so once again, we're talking about members that belong to the same species can breed together. So anything that prevents that breeding, right, is a barrier. All right, so at that point, so the populations become isolated and are considered to be, once it becomes reproductively isolated, they're considered to be separate species. Okay, so what we were talking about is what we call allopatric speciation. So we can have a geographic isolation happening within our population, and then this brings about allopatric speciation. So allopatric speciation is a mode of speciation that starts when an ancestral population becomes separated by a geographic barrier. So we can say the inhabitants of the Galapagos Islands are an example of allopatric speciation. So a small population from the mainland arrived on the Galapagos Islands. The population was geographically isolated from being able to breed with the parent population. All right, they're not going to go all the way back again just to breed. And this caused new species to develop. We could say, and other examples, that maybe a lake dries up due to global warming. And that lake, as it's drying up, becomes two smaller lakes or two smaller ponds. And so the fish become trapped in one of those two ponds. And they're not able to reach the other pond to interbreed. So this would be a geographic barrier, the drying up of that lake. Now, to a turtle living in the lake, it's not effective because the turtle can get out and walk around and go from one pond to another. So it's not a geographic barrier for the turtle. It's just for the fish or the other organisms that cannot move from one lake to the other. So what's the geographic barrier kind of depends on the animal in that environment. Another example is the three-spined stickleback. They were cut off from the sea and were trapped in small glacial lakes in British Columbia at the end of the last ice age, and therefore they evolved into different species due to allopatric speciation. In this example here, we're showing flowers that are all the same, kind of in this river valley, and then something happened over time so that the amount of water in that river started to increase and the area became much more flooded, and that is creating a geographic barrier separating our two flower populations. And over time, due to mutations, let's say, or genetic drift, this one started to become shorter, maybe slightly different flowers, so that even if the riverbed dried up and these two were able to come back together again, they would not be able to reproduce with each other. There would be enough differences that had accumulated over time that they could not reproduce. So the key to the allopatric speciation is you got a geographic barrier, and that over time will create two new species that are unable to reproduce and make viable and fertile offspring.